Welcome to today's class. Today we are going to study about the skeletal muscle in the chapter Locomotion and Movement. First of all, before going to the skeletal muscle, which we have discussed in animal tissues, the different types of animal tissues, one should know the basic difference between locomotion and movement. This term locomotion means the kind of movement in which an organism gets displaced from one place to another place. Like for example, I am standing here now. If I move to this position, then I have changed my position. That means the displacement of the body has taken place. Now this is what is known as your locomotion. So movement of an organism with respect to its position. That means displacement in the position of an organism is known as your locomotion. So locomotion is generally shown by that of your animals, and movement is shown both by plants and animals. That means where the body is fixed and the parts of the bodies are moving, like my hands are moving, my mouth is moving when I'm speaking. So locomotion and movement is seen both in case of the organisms belonging to the animal kingdom, whereas movement is shown in case of plants, but plants do not locomote, or they do not show locomotion. So for the locomotion, the type of system which is involved in case of human beings are the two types of systems. One is your skeletal system and another one is that of your muscular system. So we are going to discuss here about the muscle fiber or the muscle cell today, which is an important part of the skeletal muscle, because we know that the muscle tissues are of three types, that is your cardiac muscle, the skeletal muscle, and that of your smooth muscle. We have discussed them in broad terms in case of animal tissue. We are here going to concentrate only upon the skeletal that is muscle. Now the skeletal muscle, it is commonly known as your voluntary muscle because it is under our will. Whether we want to move from one place to another place, that is totally under our will. Of course, the central nervous system plays a role there, but definitely it is our wish whether we are going to locomote from one place to another place, or whether the movement of the body parts will take place. And where are these muscles present? Of course, these are present in case of our hands and in case of our limbs, that is the lower limbs, that is the legs. Now the skeletal muscle which are present in our limbs, they are in the form of a compact structure, and in this compact structures are present bundles of this muscles which are togetherly connected or which are togetherly externally protected by a connective tissue which is called as your fascia, and each of such muscle bundle is known as your fascicle. Inside this fascicle, you can see here I have drawn some circular objects inside each of the muscle bundle. Now this particular unit is known as your muscle fiber or muscle cell, and this muscle cell has various it is first of all covered by an external membrane which is known as your sarcolemma. Okay, and inside it the cytoplasm is known as your sarcoplasm, and also it has the cell organelle, that is your endoplasmic reticulum inside it, which is known as your sarcoplasmic reticulum. So the word sarco, when it will be coming, you have to associate with that of the muscle cell or the muscle fiber. Now each of this muscle cell or the muscle fiber is having in its sarcoplasm various parallelly arranged filaments which are known as your myofibrils, or that is your myofilaments. Now this myofibrils, if we see them under the microscope, we will find that they have this alternate light and dark bands. Now this alternate light and dark bands are present in this myofibrils because of the presence of two types of proteins in them, that is called the actin and myosin. Now actin filaments are thin filaments. These are helically bound filaments, and that is actin filaments. filamentous structures, which we will be coming now just now. That is the structure of the contractile proteins which are present within this myofibrils, which makes it alternate light and dark bands. We will be just coming to it. So this myofibrils, as I was talking about, this myofibrils alternate light and dark bands. If we see it more closely, then in between the two dark bands there is a light band, and in between the light bands there is a longitudinal flexible fiber to which the proteins are held, that is the actin proteins are held. Now this line to which it is attached, this line is called the Z line. Okay, here I have magnified a single segment, that is from one light band to another light band. That is, if you consider this one to be a light band, this one to be a light band, and in between this dark band, so in between, as I said, there is elastic fiber, longitudinal fiber to which the actin filaments are held together. Now this one is called a Z line, this one is called a Z line. So the segment between the two Z lines is known as your sarcomere, and it is the functional unit of our skeletal muscle. Okay, so like a nervous system, we have the functional unit as neuron, the excretory system, the functional unit is that of your nephron. So in case of your muscular system, skeletal muscle, the functional unit is known as your sarcomere. And what does it comprises of? It comprises of the two Z lines, that is between the light bands. Now this light bands is made up of your actin filament, and it is also known as your I band. This light band is known as also your I band. I means isotropic, that means it is non-directional, and in between there is A band. Now this A band is comprising of your that is a protein which is called as your myosin. So actin and myosin, actin forms the light band and myosin forms the dark band, and in between the light band there is a longitudinal fiber, I said. Now this longitudinal fiber represents a Z line, and this longitudinal fiber represents a Z line. So between the Z line, this unit is the functional unit which is called as your sarcomere. Understood? Now if we talk about the A band, which is an isotropic band, which is made up of your myosin, now in between this A band also, that is a myosin band also, there is a longitudinal line or longitudinal fiber which is elastic, and to which these that is the myosin fibers, or the parallelly arranged myosin fibers, they are arranged, and this line is known as your M line. Understood? So Z line in between I band and M line in between the A band. Okay, now in the resting condition, when there is no excitement of the muscle fiber, there is no movement and all, in the resting condition what happens is that this sarcomere is made up of that is your of course it has alternate light and dark bands which we have seen, but here we have the I band which is having half of the I band in this particular sarcomere unit from this Z line, and half from here of this Z line. Okay, and the complete A band is there. So if we write it in formula wise, it will be your half of your I band plus total of your A band plus again of your half I band. So this entire unit will be what is present in between this the two Z lines. Okay, and this is, I told you, this is your isotropic band, this light bands, and the dark bands are your anisotropic band. Now the structure of contractile protein, let us come to this. The light band is made up of actin filament, or F actin it is known as. This filament is, this is helically, two filaments are there which are wound helically. Helically means like this crisscross, like the DNA model, the model is helical. So same way this that is your actin filaments are helically arranged, and this single unit of F actin, that is filamentous actin, is known as your G actin, that is globular actin. The circles, this represents the globular actin. So various globular actin, they form a chain like structure, then they form the F actin. Fine, and also they have two more myofilaments which are wound around this F actin, which is known as your tropomyosin filament. Now this tropomyosin filament and the F actin filaments, they are wound with each other in the actin filament, and also there is a complex protein which is called troponin at certain distances, between certain gaps. Now what is why this troponin, or what is the function of this troponin, is that troponin masks the position in the actin filament where the myosin filament will come and bind in case of any electrical impulse, so as to show contraction of the muscles. When the electrical impulse is reached from the central nervous system through the motor neuron to the neuromuscular junction. Neuromuscular junction means where the neurons terminal endings will be, in touch with that of the muscle, that is the skeletal muscle, that is the sarcolemma of the skeletal muscle. That position is known as your motor plate, or that is the neuromuscular junction. So what happens that we will be coming later on, that how does the contraction and the relaxation of the muscles takes place, or the working of the muscles takes place, we will be coming to it. But let us now see about the myosin structure. Now the myosin structure is again, it is also made up of the things which you can see here, that is, it is called meromycin. This single unit is called meromycin. It has a head position, and the head is very important in the sense because the head acts as a ATPase, ATP synthase. That means it can synthesize ATP, it can break down ATP. So both the functions are present in the head region, that is ATP binding site is present there, and also it has two more actin binding sites. Now when will this actin binding sites will be coming and binding with that of the actin filament, is that when this troponin which is present over there, which is masking the position where this myosin will come and bind, that will be released. Understood? When it will be released, that I will be telling you. So this is the structure. This position is known as the cross arm containing the head, and this is the neck position, and this position where the two filaments are winding around each other, this is known as the tail unit. Okay, so this is the structure of the myosin filament, and this is the structure of the actin filament, and totally these are the structure of the contractile proteins. Now let us discuss about the muscle contraction mechanism. Now the muscle contraction mechanism is commonly known as the sliding filament theory, upon which the system of the muscular contraction takes place in our body. I am talking about the skeletal muscle. So we have so far come to know that the myofibrils inside the sarcomeres are there. Sarcomeres have the alternate light and dark bands, and this light bands are made up of your actin, which is in turn made up of that is F actin filaments, the tropomyosin filaments, and the troponin complex proteins, which are masking the places to which the myosin will otherwise be binding when it gets a signal from the motor neuron. Okay, and this we talked about the myosin and about the various head position, about that is your cross arm position, which will be forming the cross bridge with that of the actin filament and that of the tail position. Fine. Now what happens is that when the central nervous system by means of the motor neuron gives a signal to the muscle which is in contact with the terminal endings of this your neuron, it releases a chemical substance we all know, that is called the neurotransmitter, which is acetylcholine. Now when this acetylcholine comes in contact with the sarcolemma, that then there are certain zones, T zones, through which it enters, or it enters into the that is the sarcoplasm. So once it enters into the sarcoplasm, it reaches to a position where it is in the periphery of that of the sarcoplasmic reticulum. So the sarcoplasmic reticulum gets a stimuli, or this electrical impulse which is received by the sarcoplasm. It passes the stimuli to that of the sarcoplasmic reticulum, and it releases calcium ion into the sarcoplasm. numerous calcium ion it releases into the sarcoplasm. Now this calcium ion, what does it do is that it comes and it binds with that of the troponin, which is masking the area to which the myosin filament will be binding otherwise. So once it comes and it binds with that of the troponin gets, because it's a protein, so unmasking of that region takes place. So binding of the calcium ion with that of the troponin will release the troponin from these areas which are now released, so that the myosin filament can easily bind with that of the actin filament. Now the myosin filament, once this troponin is released from this that is your actin filament with the help of the ATP, because here in the head region we know that there is ATP binding sites. Now this head region will utilize the ATP, so that now the head region which is having the actin binding site will go and bind with these locations where otherwise was troponin in the absence of the calcium ion. So once it binds with that, then it will contract the muscle. It will slide the actin filament towards itself. The myosin filament will pull the actin filaments towards itself. As a result of it, what will happen is that the Z lines, that is these two lines which are the two endpoints of a sarcomere, they will contract. They will come closer. So once they will come closer, these two things will be coming closer. Now you can see here, these are the I bands. So the I bands are flexible, they are thin, they are flexible, they are made up of this actin filament. They come closer. So they come closer, but this A band, which is made up of your myosin, it remains as it is. There is no contraction of this, but the contraction of the I band takes place, bringing the Z line together. So that will result into the contraction of the muscle and the movement of the concerned organ, whether it is hand or leg or whatever it is. Okay, and now the next thing what will happen is that now the ADP and the inorganic phosphate, which is formed as a result of utilization of ATP, will be released. The myosin will now relax, and now what it will be doing is that it will take another ATP in the ATP binding site, and the cross bridge which was made between that of this actin filament and with the head of this myosin filament, there will be a breakage. Understood? So there will be a breaking and this will be released, and this will keep on happening until and unless the calcium ion again is absorbed back by the sarcoplasmic reticulum. So once the calcium ions are taken up by the endoplasmic reticulum present in the, that is your sarcomere, that is that is your myofibrils, then what will happen is that there is again a deficiency of your calcium ion. As a result of this deficiency of the calcium ion, now what will happen is that again the troponin will be masking those areas which otherwise was the region where the myosin would be holding to that of the actin filament. And since the actin filament is sliding over that of the myosin filament, that is why it is known as your sliding filament theory. Understood? Now this is actually the principle, or this is actually the mechanism, that how does the muscular contraction takes place by the help of this actin and myosin in presence of ATP. ATP is required twice. Once it is required when the starting of the contraction process takes place, that is in presence of ATP only, this myosin will be now going and binding with that of the areas which otherwise was taken up by the troponin. There is an attraction, attraction between that of the actin filament and the myosin filament together. Actin at the top and myosin is somewhat broader and somewhat fixed, so from between them there will be an attraction. So it's like two magnets having the two opposite poles, they will be pulling with each other and they will be slide pasting each other. Okay, so once their ATP is utilized, and secondly ATP is utilized when the contraction is over. Now the myosin wants to relax, it wants to break down the cross bridge, the cross arm wants to release itself. So how will it release? Again with the help of ATP, the releasing of the bonding between that of the actin filament and the myosin filament will be broken. So this is how the mechanism of the skeletal muscle takes place, that is a voluntary muscle or the striated muscle as it is commonly known as, because of the alternate light and dark striations present within the muscle fiber, which helps in the movement or the locomotion of an organism. Now there is a pigment which is called as your myoglobin. Now those muscle fibers which have this red color pigment more in them, they are called the red muscles, okay, red muscle fibers. And those who have less number of this myoglobin pigment in them, they are called as the white muscle fibers. Generally the red muscle fibers have more mitochondria in them, and they are known as your aerobic muscles. Okay, they are known as your aerobic muscles. They work very finely in presence of oxygen, but when the body has to show vigorous movement, constant movement, in that case it is the white muscle fiber which is having less mitochondria. In absence of oxygen, they work. Understood? So this is the entire mechanism of the muscular movement, the two kinds of contractile proteins present in the sarcomere, about the muscle as a structure, then a single unit, and then how does it looks like under the electron microscope, and the entire working. I hope you understood the today's class well about the skeletal muscles movement and the various proteins, their structure, and the sliding filament theory, that how does the muscle contraction and relaxation takes place in our body. Thank you.